In this segment, we will discuss about the mass analyzers. We have been discussing about the mass spectrophotometry, mass spectrometry, and the different detectors which are used. Now, we will discuss the mass analyzers, what they do, what is the principle of mass analyzers. Now, as we know that once when we place a substance or a molecule in a mass analyzer, due to its formation, due to its construction, due to its principle, the electric current, it falls on it, and the ions, they are created on that molecule. Now these ions, once they are created, they leave the ion source and they pass into the mass analyzer. Because it is mainly the mass analyzer's function is to determine the mass of the ions and the quantity of the ion which passes through the mass analyzer. Now, the function of mass analyzer basically is to separate the ions and to measure their masses, as the name implies, because the different ions, they will be of different mass, and when they will pass through the mass analyzer, it will analyze them according to their masses. It will separate them according to their masses. So at any given moment, ions of a particular mass are allowed to pass through the analyzer. So analyzer, it allows the ions of particular mass to pass through it and then analyzes it. At an analyzer, the ions are then counted by the detector. As the particular ions of the particular mass, they are allowed to pass through the analyzer, and then the mass analyzer, it counts them by the detector which is present them, that how much ions of mass A, B or C, whatever it is, they have passed through it. Now, ions of different masses, they are allowed to pass through the analyzer, and again the detector counts the number of ions. Now first, the ions of specific masses are allowed to pass, whatever we set, but then in a general condition, the ions of different masses, they are allowed to pass, and they are counted by the detector that how much ions have passed through the mass analyzer. Now analyzer then, it scans through a large ranges of masses. Whatever mass of ion, whatever ions of different masses have been passed through it, what it does, it analyzes it and it scans those ions, that how much ions of this mass have passed through it. Now, in the majority of instruments of mass analyzing, a particular type of ionization is coupled to a particular mass analyzer, so that it can determine a specific mass, and the particular mass analyzer that operates by a particular principle of that instrument. Now, some well-known mass analyzers, they are quadrupole mass spectrometry, ion trap mass spectrometry, nanospray and online tandem mass spectrometry, and some well-known mass analyzers. Among these are magnetic sector analyzer, that is those analyzer which use the magnetic sector. Then accelerated mass spectrometry, that is where the mass particle, they are accelerated under a ion, under the charge. And Malditov mass spectrometry, that is the Malditov which we have been studying earlier. So we see that the mass analyzers, they are very important instrument in the molecular biology, because with that we can find out what are the different masses of different proteins, different nucleic acid, which are present or which are connecting with each other, and thereby we can find out the function as well as the regulation of the protein mechanism.